So my name is John Spear. I'm from the Department of Civil and Environmental Engineering, and we're here today for the master's thesis defense of Kevin Chan. Kevin and I met 4 years ago, and he took my second semester environmental engineering class. It's hard to believe that was 2013. Yes. Kevin expressed interest in staying here at Mines for a master's degree to go after his BS degree that he got here. So he did a non-thesis master's and was working in our lab, kind of doing some projects while he was doing that. And then we decided to have him also do a thesis master's. So he is a glutton for punishment because this is his second master's from the Colorado School of Mines. And then what made matters worse is like Kevin was getting good at doing things in the lab, and I'd walk over to Kevin and say, "Hey Kevin, we ought to do this. Hey Kevin, we ought to do that. Kevin, have you thought about doing this other project right here? And Kevin, can you now do 4 projects at once?" Right? And so he's here today to tell us about a couple of stories that he's been working on for the past year and a half or 2 years or whatever. Kevin was funded by the West Center, the ConocoPhillips West Center here on campus, which has been a great thing. And that's what's gotten you through to this point. So let's have you tell your story.
Cool.
Thank you, Kevin.
Thanks for that introduction. Thank you all for coming to my defense this morning. For the past year and a half, I've worked on the microbial ecology of activated sludge and granular activated carbon communities, assessing the resilience community structure in treatment for produced water. A small overview of what I'll be going over today. I'll start with an introduction, talking about oil and gas production within the U.S. now, as well as talk about some drivers for reuse within that industry. The 2 treatment systems that I worked on are a sequencing batch reactor, membrane bioreactor, and a biochemistry active filter. We'll go over the experimental design of each of those, talk about some results and conclusions from both of those projects, and then we'll end with some future directions from this work, coming from this work. So in the last 20 years or so, oil and gas exploration has seen exponential growth within the U.S. This is a graph by Mangosh et al. Showing different basins throughout the U.S. And the amount of production coming out of those basins. So in the past few years, particularly the Haynesville and Marcellus basins here, the production has almost quadrupled since 2010. So it's a really big growing industry. Locally, the U.S. Energy Information Administration collects data on the amount of production wells in Colorado, and that same trend is also seen locally in Colorado as we're getting up to about 50,000 wells or so currently. And all these advancements in hydraulic fracturing and the technology have really allowed the U.S. to become more energy independent, which is great. So what is the process of hydraulic fracturing? It is the injection of water under high pressure into bedrock formations to allow the flow back and collection of oil and gas flow from the subsurface. This process has been done since the 1940s, and within the U.S., we've fractured approximately over 1 million wells. Globally, that's about 2 million wells total. Production today, about 95% of the wells in operation are hydraulically fractured, so it's very common practice in the industry. How does it work? Injection fluid containing about 90% water, 9% sand, and about 1% chemical additives are pumped into the subsurface. The high pressure in the water fractures fissures in the bedrock. The sand keeps those fissures open. The flow is reversed back up to the surface, and oil and gas can flow back with the water up for collection. 2 waste streams are primarily generated during the operation. The first is the frac flowback water. That's primarily the injection fluid that operators pump down into the subsurface. After a while, it changes into what's called produced water. And this is the water that's native to the formation that's already there. Our research is focused on the treatment of this second stream of produced water. The water used for fracturing is typically sourced from surface and groundwater resources locally. A single well can utilize about 2 to 16 million gallons of water over its lifetime. The equivalent of that is about the production of Golden Water Treatment Plant for our citizens here in Golden. In the wintertime, we produce about 2 million gallons per day. In the summertime, it's about 16 million gallons per day through that plant. The recovery of water when we reverse the flow back up from the subsurface can vary as the formation characteristics change. but typically it ranges from 10 to 80% recovery. Produced water is characterized as a high brine solution. It's very salty, very high in salt, TDS content, and that makes challenges for its disposal as a waste stream. Public treatment works and landfills generally do not accept produced water because of this high TDS content, because the salt tends to kill microorganisms vital for their processes, such as methane generation in landfills. Therefore, deep well injection is a very favorable process for the disposal of this waste stream because it's more economically favorable in a lot of regions. However, this eliminates storage within our modern water cycle because the recycle rate back up from deep well injection is back up to the surface is on the order of millennia. This is a graph showing the western United States along with the location of hydraulic wells and the water stress associated in those regions, particularly here in Colorado, the square there, and then Texas, kind of this lower bottom region here. There is a high density of wells associated with extreme water stress in those regions. Deep well injection in these areas really starts to exacerbate water shortages in the local level. Zooming in a little bit more specifically, again, Colorado and Texas here. About 50% to almost 100% in Colorado, those wells are under areas of extreme water stress. The current pathway for produced water is, again, deep well injection after some treatment, maybe not even any. But with the implementation of advanced technologies, we can increase the beneficial reuse of this waste stream. That can help to augment stream flow, be used for other irrigation processes, as well as internal reuse within industry itself on other hydraulic fracturing operations. In our studies, we utilize produced water from the Denver-Julesburg Basin in northeastern Colorado. These wells produce about 3 million gallons per day of produced water, and the typical recovery in this basin is about 50%. The water quality characteristics of the produced water are shown on the right here. As mentioned before, produced water is very high in salt content. And the DJ Basin is sitting roughly about 23,000 in total dissolved solids in the bottom, milligrams per liter. Other basins can range from lower to that to as high as 300,000, which is about 10 times greater than seawater. The first treatment system that we worked on is a sequencing batch reactor membrane bioreactor, or SBR-MBR, that was designed and operated by Victoria Frank. This was operated at the Mines Park apartment complex for approximately 1 year. The project is aimed at the co-treatment of municipal wastewater with produced water. Throughout the experimental phase, we increased produced water additions into the influent over time and monitored the microbial effects, or shifts in microbial community, and correlated those with the system's performance. Our research questions for this project include: is the dual exchange treatment with residential wastewater viable for produced water reclamation? And are the activated sludge communities resilient to produced water additions? According to the EPA, only the effluent from systems destined for environmental release or drinking water augmentation is regulated. Other reuse requiring pretreatment, those standards are more process and industry specific. We set our treatment goals based on the Clean Water Act and Safe Drinking Water Act by the EPA to get nitrate below 10 milligrams per liter, and then have our COD in the effluent, or chemical oxygen demand, between the range of natural waters and raw sewage. A process flow diagram for SBR-MBR. We started with 2 isolated tanks, a municipal feed tank and a produced water tank. Those 2 streams were mixed prior to the addition to 2 bioreactors. They flowed in upflow configuration in the parallel bioreactors. Clean water was decanted off the top of these reactors into an ultrafiltration membrane on the right here. This was mainly used to decrease turbidity until coliforms in the effluent that permeate from this membrane tank was eventually wasted back into the municipal sewer lines. The targets represent our biological sampling points throughout the system, and those will be correlated with phylogenetic data later on. Our system was designed to treat 200 liters per day and maintain internal temperatures about 14 to 20 degrees C. Both of the bioreactors were seeded with activated sludge from a demonstration scale SBR-MBR that was already effectively treating municipal wastewater. Each reactor went through identical 2-hour treatment cycles that include filling, settling, and aeration stages. The reactors were offset by 1 hour from each other in order to ensure continuous operation and separation of those different stages between each one. We had 5 experimental phases throughout this project. We started with baseline testing with just residential wastewater and 0% produced water additions. We slowly ramped that up to 6% and eventually 20% later on in the experiment. After the 20% addition, we noticed a loss in system performance. I'll talk a little bit about that a little later, but we decided to ramp the additions back down to 6 and 20 to assess maybe a potential recovery of the system's treatment. For microbial analysis, we filtered our biological samples through 0.22 micron filters. We extracted DNA using a mobile power soil isolation kit and quantified that DNA using qubit broad-range assays. We used the Illumina MiSeq platform to do DNA sequencing, and then bioinformatics software was used, like QIIME and R was used to analyze the results that we got back. A principal coordinate analysis was constructed using the resulting sequences, and what's unique about this is the microbial diversity seems to be more dependent on the location within the system, as you see 3 different clusterings of each influent water and then the bioreactors. It is interesting to note that we do assume that the bioreactors are going to be the same, and we do see that in this PCOA plot, as the bioreactors do cluster together with a insignificant p-value and differences. This is phylogenetic data from the residential wastewater feed stream. We only present up to the second 6% addition of produced water, only because we could not get quality sequences in the last one, but we could infer what those communities would be like using the previous phases there. A key point about this data is that the influent community from the residential waste stream is pretty stable. There's not a lot of changes here, and so this cannot be a variable for change in performance later on in the system. In the community, we see a dominance of proteobacteria, which see a dominance of the phyla proteobacteria, as well as firmicutes, microorganisms common to wastewater streams, such as the order Clostridiales, which is within firmicutes, really agree with other, occurred within the influent stream, as well as an increased organic loading from the apartments. And this can be verified when we look at our TDS data, or salt content. In phase 1, we see at the very end of phase 1, we see an increase of salt. Flavobacteria are very halotolerant, and they can proliferate in high salt environments. Phase 1 also occurred during the summer months of the academic year, and so as we get into the 6% and 20% additions, more students are in the apartments contributing to that wastewater, increasing the organic load, which may help to proliferate, or may help the proliferation of flavobacteria in the influent stream. Compared to the produced water stream, we do expect this to be different because of different environmental factors such as temperature, pressure, and other nutrient content in the subsurface. This community is dominated by proteobacteria and Bacteroidetes. Flavobacteria are very common in this stream because of that high tolerance for salt, and also they've been associated with the degradation of hydrocarbons, which are a part of oil and gas waste streams. The bioreactor phylogenetic data is presented here, the bioreactor 1 and 2. Again, we expect the communities to be the same, and we tested that with Anderson statistics and got back an insignificant p-value to confirm that observation. Originally, in the residential wastewater influent, we saw a dominance of firmicutes. Here, we observe a decrease in the abundance of firmicutes. This is most likely due to the aeration process within the bioreactors, introducing oxygen, which is toxic to firmicutes, which are mainly anaerobic bacteria. We see a growth in more halo-tolerant organisms within the bioreactors with the 6 and 20% produced water additions. Flavobacteria and chloroflexi are microorganisms that are more tolerant for salt content. It is interesting to note that in terms of performance of the system, we don't see any variations in organic removal. So this means even though we see some changes in abundance of certain microorganisms within the community, overall function is still stable. We're still able to remove the amount of organics that we need to. However, this trend is not seen when we look at constituents such as nitrogen or ammonia removal. That data is presented here. Ammonia percent removal is on the left there, and then we have the different phases of produced water additions. Right around the 0 to 6%, we get almost about 100% removal of ammonia. But when we go to 20%, that removal starts to drop drastically. During the recovery phase that we wanted to analyze, at the 6%, we get system recovery and then another drop off at the 20%. So it seems like the salt content may have a big impact on the community structure. Something's happening that's affecting the ammonia removal. When we look at our phylogenetic data during these time points, more specifically nitrifying bacteria, we see a similar trend. So key organisms for nitrification, which are Nitrous ammonitalis and Nitrous spiralis, which are the key ammonia and nitrite oxidizers, respectively, we see a decrease in their abundance right around that 20% removal rate. With that initial increase in percent removal, we see an increase in their abundance to about the 6% range. But then the salt content really starts to affect their abundance. Nitrous ammonitalis has a very low or very limited tolerance for environmental shifts, so their decrease is most likely the cause of nitrification loss in the system. It seems like at 6%, the function of the population recovers, but it doesn't seem like their abundance does. We look more closely at these sampling points. These are early on in the 6% phase, and so we expect the abundance of these organisms to increase along with that recovery and removal. Some conclusions from this study, just to restate our research questions. Is dual stream treatment with residential wastewater viable for produced water reclamation? With our results, we say yes, it is. We are able to get sufficient organic and ammonia removal below the regulated amounts of regulated concentrations as set by the EPA. And are activated sludge communities resilient to produced water additions? Yes, they are. Bioreactor communities were able to adapt to that 6% produced water addition. The 20% was a little too much stress on them, but they were able to recover when we ramped the additions back down. An additional conclusion is that we found a threshold of nitrification levels between 6 and 20% produced water additions, indicating there might be an optimal fraction between produced water and residential wastewater. Some lessons learned from this study: the activated sludge communities are resilient and they are able to adapt to dual stream treatment configurations. We do observe nitrifying bacteria inactivity associated with an increased TDS or salinity concentration. So it seems like salt can really negatively affect the community. So we wanted to take a look at some pre-treatment options maybe before this SBR, MBR that can help make the beneficial reuse of produced water more attractive. One of the most common techniques is membrane desalination. However, this technology comes with its own issues, particularly organic fouling in the membranes, decreasing its lifetime and performance for reducing salt content. So what could we do to reduce the organic loading of the influent coming into those technologies? We wanted to look at a biologically active filter, which is our second system. This was designed and operated by Stephanie Riley from Aquatech. We focused on monitoring the microbial development within these columns, and the system was configured based on 3 different phases, and we evaluated the COD removal as a parameter of pre-treatment for desalination processes, really tackle that bio load before membrane desalination. This was operated with fresh granular activated carbon media, and this makes the system unique because although there might be some microbial communities within the GAC, there's not an established functionality within the system, and so it's unique because the native communities to produce water are seeding this media filter. Our research questions for this project are: is biological filtration with granular activated carbon effective pre-treatment for produced water desalination? And then, does the microbial community structure direct system performance? A process flow diagram for the BAF. Configuration A, what occurred during phase 1 and phase 2, they are the same. This had an oxo column and anoxo column in parallel with a single feed tank that contained produced water. In phase 3, we went to configuration B, which isolated the columns separately as 2 different treatment systems entirely. The flow into the GAC beds was in an upflow configuration. We aerated the oxo column with about ½ liter of air per minute. The flow rate through the systems was about 0.8 gallons per minute, and then we wanted to backwash to prevent clogging of the filter with tap water, and we were able to achieve about 50% bed expansion with those. The phases are dictated by operational shifts. At the end of phase 1, we observed a pretty big clogging event in an oxo column. We had to fix some of the piping, remove the clog, and replace some of that GAC media material. The end of phase 2 was that isolation of the columns entirely. It was 2 separate treatment systems. Our microbial analysis was the same as our SBR, MBR techniques, except for we have the addition of using ATP as a proxy for biological activity within the system. That was measured using the Promega bacterial glow assays. Our phylogenetic results for each of the columns, oxo column on top and anoxo column on bottom, are shown here at the class level. One of the interesting observations that we make is the oxo column seems to increase in diversity quicker than the anoxo column. This is expected because of the addition of oxygen throughout the environment. The anoxo column increases diversity right about the end of phase 1 with that clogging event, and we suspect that this is because of the inability to backwash the system anymore. We allowed the microbial community to sit and proliferate for a while instead of backwashing, which would have removed some of the growing communities and not be able to establish as well. In phase 1, for both of the columns, we see a dominance of gamma proteobacteria. This agrees with previous research, as they are more halotolerant organisms, such as Shionella and Halomonas. In phase 2, the diversity in both columns start to increase. Microorganisms common to produce water reservoirs, such as Rhodobacterias and epsilon proteobacteria, start to grow in both of these systems. During phase 3, we start to see an increase in delta proteobacteria, more specifically Drosophila monothales. These are obligate anaerobic bacteria that cannot survive with oxygen. And it's very interesting to note that we see their abundance increase within the oxic column. We suspect that this occurs because of different microgradients within GAC. The outside of the media granules could be oxic, but oxygen is not able to penetrate further into the granules themselves, causing an interior anoxic region. It's also interesting to note at the very end of phase 3, the communities start to look similar between the oxic and anoxic column, and this can be confirmed with a principal coordinate analysis using a fixed axis with time. During phase 1, we see a separation in diversity between the oxic and anoxic column. But over time, in phase 2 and 3, those start to converge together to confirm our previous observation. Interestingly, these effects during phase 1, such as the separation and diversity, aren't seen in system performance. In terms of COD % removal, which is graphed here against time, in phase 1, oxic and anoxic column perform similarly. This removal rate remains pretty high through phase 1 and halfway through phase 2. In the transition to phase 3, we see a decrease in the capability to reduce COD within the system. We suspect this is a relationship between GAC sorption as well as microbial activity. Right around the transition period from 2 to 3, we may have lost some of that activity, and the GAC sorption processes aren't able to effectively treat COD to the same extent as they did during phase 1. This can be confirmed with our ATP analysis, graphed here in micromolar over time. In phase 2, we see really a recovery, an increase in activity correlated with that high COD removal. However, in phase 3, it starts to taper off, and that's right about the same time we see the decrease in COD removal. So it seems like there is a relationship between the microbial activity and performance of the system. To conclude this study, to restate our questions, we want to know, is biological filtration with activated carbon an effective pretreatment option for produced water desalination? And with our results, we do agree that it is possible. We're able to achieve about 55 to 90% removal of COD in the effluent. Our second question, does the microbial community structure direct system performance? With the ATP data, we would agree that it does. Once the microbial activity started to diminish at the end of phase 2 and 3, the GAC sorption processes were insufficient to keep the COD removal as high as we expected earlier on. An additional conclusion is that the oxic and anoxic communities converge in diversity over time, and we're still able to achieve similar system performance. So regardless of if there's oxygen or how the system is brought up, they do converge on a similar community, similar system performance. To summarize, both of our treatment systems, we found the microbial community plays a pivotal role in determining system performance. As in the SBR-MBR, we noticed a loss in nitrifying bacteria correlated with a loss in nitrification and lower removal of ammonia from the waste stream. In the BAF, we found a loss in biological activity correlated with a lower removal of COD within the system as well. However, we did find that both of these treatment systems are actually successful for the treatment or pretreatment of produced water. And by understanding the phylogenetic makeup within these systems, we can optimize these processes further and make them more efficient. Combining these two systems could also lead to a potential fully encompassing treatment train for oil and gas produced water treatment. This combined with membrane desalination could allow for the beneficial reuse of this waste stream, such as environmental release or even drinking water augmentation. Some future directions that can come from this work: we would like to experiment further with the SBR, MBR, really find that optimal fraction of produced water to residential wastewater that's able to sustain adequate performance of the system. It would be interesting to do a GAC granule autopsy to analyze those microgradients we discussed in the oxidant column, really see what processes are going on within a granule. It would be pretty valuable to adapt these systems to other produced water sources with different water quality characteristics, really test the flexibility of these systems to compete with other disposal methods. And then doing an economic analysis of a full treatment train compared to deep well injection would be really helpful to try and make beneficial reuse of produced water more attractive rather than the traditional method of deep well injections. I want to thank my funding source, the ConocoPhillips Center for a Sustainable West, for funding our research. I want to thank my committee, my two advisors, John Spear and Zahi Kath, my committee members, Junko and Chris, for really believing in me to be able to do this work, getting me excited about doing this sort of research. I'd like to thank people from the GEM Lab and our micro lab, Dr. Josh Sharp, Chris Trevetti, Zach Jones for really mentoring me, helping me out, getting accustomed and acclimated to the lab. Holly for editing my thesis, comforting me when I start to get stressed out. Other members, Stephanie Carr, Tess Weathers, Blake Stamps, Gary Vanzen, Dina Drennan, Kristen Mickelson for really mentoring me throughout analyzing my data, learning how to be more of a microbiologist. Chelsea Bachman and Emily Krause for helping me with bioinformatics, and really everyone for keeping my sanity throughout my thesis journey. I want to thank people from Aquatech, Victoria Frank and Stephanie Riley for designing and operating these systems. I had no idea how to do it, and I'm glad that you were there to help me really keep them going. Stephanie Bustos, Danica O'Hore for running water quality analysis and doing ATP, helping me finish those samples up, and Dottie Ramy for mentorship throughout with some of the water quality characteristics and analysis there. Finally, I want to thank a lot my friends and family for keeping my spirits up, being there for support, saying it's okay to sleep over in the office and not come home. I just want to thank them for all their support. And thank you all for coming today. Take any questions that you may have.
Take questions from anybody.
Yeah, Blake.
So you sort of talked with the BAF. What would you do to change, like in stage 3, where it seems like everything dies? I mean, is there something that you could do to stop that?
I think diving a little deeper in the phylogenetic data would really help with that. Maybe doing some metabolic analysis. I think also the autopsy with GAC granules will help. Maybe we're not aerating the columns enough to get that oxygen penetration within into the interiors of those granules. Those might be valuable in really assessing what can be optimized in those systems. Yeah.
Kevin, when you were doing the work on the SBR, MBR,
yeah.
and you saw the reduction in the microbial community that oxidizes nitrite, nitrate, was that reflected in the effluent when you were looking at the ammonia and nitrates and nitrites? Did you see a correlation in the species of nitrogen in the effluent that you could match up with your phylogenetic?
Yes and no. We mainly looked at ammonia removal. So taking a look at the symbiotic relationship of those nitrifying bacteria, if we see an increased amount in ammonia, that means the first step is not functioning correctly. So we would expect the species in the effluent to decrease because the downstream reactions with other bacteria, such as the nitrite oxidizers, they're not able to perform when the primary reaction rates aren't happening with other biological processes. So I guess I don't know the species, the different counts of the compounds of nitrogen in the effluent, but based off of our results from ammonia, that's what we can infer.
Could you please go back to page slide 18? I didn't quite understand those. Explain it again.
Yeah, this is a principal coordinate analysis, which is a statistical method for testing the similarity of microbial community structures within different time points. Really, the takeaway from this is that there are 3 clusters. So they cluster together. That means they are more similar to each other. There's 3 very distinct clustering of each of the influent waters and then the bioreactors. This was really used to just confirm our theory that the bioreactors should be the same because they were operated under the same conditions, they were seeded the same, things like that. Does that help? Yeah.
Building on that question, for this one, clearly you sequenced the water. Did you ever sequence the DAC itself, or do you always sequence water coming out of the filtration system?
In the filtration system, we sequence the GAC. Yeah, we sequence the GAC instead of the water.
And so, building upon this sort of analysis, did you ever do an analysis to see how easily in your produced water GAC would compare to what was in other sort of GAC systems that they would derive from the same? Because I mean, this was derived from a plant or
This was actually fresh GAC.
so it's fresh GAC.
Yeah, it was not used in any previous treatment trains at all.
Did you ever look to see if the community was similar to something you see in, say, straight water treatment type water? Or I guess the question is how much does salinity skew the community from water that might have everything but the salinity?
In comparison, we didn't compare that to like traditional GAC media filters.
What do you think?
I think we would see a very different community. In ours, we saw a lot of halotolerant bacteria that are also able to perform those biological processes for removal. So we could see really the same function of a system, just performed by a different community. So really the flexibility within these types of systems for different water quality influence coming in.
Any other questions? Okay, Kevin, thank you very much.